I think you are all fit and fine. Today we will learn a, a very important topic, that is color blindness. Color blindness. You know that color blindness uh, is a genetic disorder that happens for the chromosome, the default chromosome of X chromosome. We know that in human body there are two types of chromosome, autochromosome and sex chromosome, and in female body there are X plus X chromosome, and in male body there is X plus Y chromosome. And if there is a problem of X chromosome, and for this one in our eye some protein does not do not produce, and for this kind of problem a person can't make difference between the colors of red, then uh, green and blue. So today's topic is very interesting topic, and we are starting this one. To know this one, we should learn about the structure of eye. We know that if we cut our eye this way, we can find that our eye has this structure, and most of the time you have seen also this structure. Actually, the first layer, the outer layer of the eye, that is called sclera, and the second layer that has melanin, this layer is called choroid, and the third layer, the most innermost layer, that is called retina. And we know that here some the choroid comes for this our eyeball mix, here the blackish part, and this one there is a gap, and this gap is called the pupil. Light comes, enters in our eye uh, with this hole, and there is suspensory ligament, you know that, and there is lens in our eye, and there is a liquid that is called vitreous humor, and in front of this there is also liquid that is called aqueous humor. So we will discuss this topic descriptively in another class. Today we are going to learn the color blindness. Actually, this retina, it is actually nerve cell layer. It has full of nerve cell, and it has three layer. First, the innermost layer, that is called the ganglionic layer, then bipolar layer, and the outermost part, that means this part, this is called the light reactor area. Light reactor area. In light reactor area, there are two types of cell. One that is called rod cell, and another that is called cone cell. Understood? First one that is called rod cell, and another is called cone cell. Actually, this rod cell is named for the shape of it. Rod cell means uh, they has the two part and both sides are rod like structure. Rod cell. I have no space to draw. I am drawing here. Rod cell, they have two part, and their structure is like that. And cone cell, that has two part also. First, the lower part, that is rod like structure, and the uppermost part that has the cone like structure, so that this one is called the cone cell. And cone cell are made of some protein, opsin protein. They are made of opsin protein, and they has different type of opsin protein. In rod cell, the protein that has this is called rhodopsin. Ro, rhodopsin, rhodopsin protein. And in cone cell, there are three types of protein. There are three types of protein. First one, that is called the erythropsin. Erythropsin. And then chloropsin. And then cyanopsin. Cyanopsin. So these three types of protein actually attracts and collects the light. We know that uh, red, green, and blue, they are the main color. They are called the primary color, and with the help of this color, the other colors are made. So, if all the protein we have in retina, especially in the cone cell actually remains active in the daytime, and rod cell becomes active in at the light, very night time actually, or evening time. That means when the light has very small amount of light. And cone cells is active in the full light area, that means daytime. So if a person has all this protein, that means erythropsin, chloropsin, and cyanopsin, then all the cone cells are active, and he can get all the lights, and he or she can differentiate between the colors. But if any of the proteins, erythropsin, then chloropsin or cyanopsins, if there is absent in any human's body, especially in the retina, then this color cannot be captured by the light reactor area. So that the problem happens. And these areas, these proteins are supplied by X chromosome. So it is X linked. So if a person has not erythropsin, then he or she can't get the red light. This his or her retina is unable to capture this light. If anyone has not the chloropsin, then he is not able to capture the green light. And if anyone has not the cyanopsin, he is also unable to capture the blue light. So this problem happens, and these types of people are not able to make differentiate about the light, especially the three colors: green, red, and blue. This problem is called the color blindness. Actually, this is the color blindness. This is X-linked problem. And most of the time, we can see that in the world there are 8% male, 8% male, they are attacked by the color blindness, and female are very less, only 0.04%. That means out of 1000 female, only 4% is attacked by this problem. Why? Actually, this happens for their chromosome. In male organism, we know that there are X plus Y chromosome, and in female X plus X chromosome. If the male's X chromosome, only one X chromosome, and that is faulty, that are not able to produce the erythropsin or chloropsin or cyanopsin or both, then this male person cannot catch these types of light, and he is in color blindness problem. But female has the double opportunity, 2 time X. So if anyone's first X is defaulty, then it may be the other chromosome be very rightful, and this other chromosome has no any problem. So she is not unable to capture the light. And if he is very bad luck, she has very bad luck, then both the X chromosome will be faulty. So this case is rare, so that only out of 1000, only 4 female are attacked by the color blindness. So I think you have learned well.